Okay, so today we're going to take a look at Chapter 3, Amino Acids, Peptides, and Proteins. We've got two learning goals for Part 1, which is to look at the structure and naming of amino acids. And then we're going to start looking at the structure and properties of peptides that result. So if we take a look at the topics that are coming up for Exam 1, just that everyone's on the same page, you're going to be responsible for unit conversions, hydrogen bonding, as well as molarity and osmolarity. And then you're going to see some questions about solubility and ionization state, as well as the anion gap. And we're going to explore in detail pH and buffers, as well as what we're going to talk about today, the properties of amino acids, calculating the PI, the isoelectric point of a short peptide, and then an ability to explain how to separate or isolate amino acids, including things like 2D gel separation, and then the ability to predict what elution will look like from column chromatography. Today, we're going to focus on proteins, looking at the molecules that are the main agents of biological function. So proteins have a variety of roles, whether that's catalysis or transport or structure or motion. And really, the variety of proteins is pretty incredible. I mean, if you think about it, it's everything from enzymes to hormones to antibodies to transporters to your muscles to the lens of your eye to feathers to spider webs to horns to milk to antibiotics to mushrooms, poisons, luciferin, hemoglobin. Really, proteins run the gamut. So significantly different molecules based on their amino acid sequence. So let's take a look at that. We know that amino acids are monomers, and we string these together to make our proteins or our polymers. And it's probably useful at this point to say a couple of things. Proteins are the most abundant biological macromolecules, and because of their diversity, it's really quite incredible when you look at the fact that there are really only 21 proteogenic amino acids. So we have the 20 of our standard genetic code, and then we have selenocysteine. And I think what's pretty striking about this is that humans can synthesize 12 of these from each other or from other molecules within metabolism. The other 9 have to be consumed. So usually they're consumed within their protein derivatives, and that's why we refer to them as the essential amino acids, and those are going to be histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So let's go back and take a look at what we know about amino acids. We know that they share many features in common. So for example, you have this NH3 with a positive charge. You also have this COO minus, the deprotonated carboxy, and the amino terminus. Then we have this alpha carbon. So this is the carbon at which our R group is attached. And what we're going to see is that we have 20 different R groups. So with the exception of proline, everything else in our molecule is the same. But it's really this R group that provides the striking differences in terms of the behavior of our amino acids. It's worth noting that most alpha amino acids are going to be chiral. So that alpha carbon is always going to have 4 substituents. And because of that 4 substituent, it's going to be tetrahedral. With the exception of proline, all are going to have an acidic carboxyl group, a basic amino group, and then usually that alpha hydrogen connected to the alpha carbon as well. And it's this 4th substituent R that's unique. So in glycine, the 4th substituent is also going to be hydrogen. So if we take it what you know about nomenclature and naming, we're going to go back to your R and S. And we're also going to talk briefly about D and L. So glyceraldehyde is the standard. And so for D and L, the enantiomer that is rotating plane polarized light clockwise is designated as D. And the other enantiomer, the negative, because it rotates plane polarized light counterclockwise, became L. And so you're going to need to be able to distinguish between R and S as well as D and L. And because all amino acids are chiral with the exception of glycine, it's worth noting which one, whether it's D or L, is most common. So proteins that we see in life tend to contain only L amino acids. So in this case, that means they have the S configuration. So if you take a look at L-alanine, You can actually see the structure there in terms of what the orientation is of the carboxy and the amino terminus. And we can actually see here then, right, our Fisher representation, as well as the ball and stick model, and then what we would probably be most likely to draw in terms of the structure of that molecule. Now, when we're looking at classifying amino acids, it's useful for us to think about how we might group these. So common amino acids can be placed into 5 basic groups, depending upon their R substituents. So they could be nonpolar and aliphatic, right, containing only carbons and hydrogens. They could be aromatic. They could be polar and uncharged. They could be positively charged and negatively charged. And one of the reasons why we want to think about this grouping is because it really truly is structure that determines function. So if we take a look at the structure of these molecules, we're going to be able to know something about how they're going to behave. One of the tools that we use to examine this is the Lambert-Beer law, so spectrophotometry. So this is where absorption is equal to epsilon Cl, where the C is our concentration, L is our path length, usually it's 1 centimeter, and epsilon is this wavelength-dependent molar absorptivity coefficient that has the units of our concentration and path length, because absorption is going to be unitless. So how do we do this? Well, our experimental measurements are usually made in terms of transmittance, which means I shine a beam of light, and I have my initial light intensity that's coming out of my spectrophotometer, and then that sample of light is absorbed by our concentration of whatever liquid we're using, and then what comes out is what I measure. So the light intensity I is what's coming out of the spectrophotometer. So I can relate those two in terms of transmittance, where T is equal to this ratio of I over I naught, and absorption is going to be equal to the negative log then of T. Now it's useful to have a conversation about some of the limitations of this law. So it's going to be limited both in terms of chemical and instrumental factors. So it can become nonlinear, right, if any of these conditions are met. So if the concentration is too high, it can result in electrostatic interactions. If there's scattering of light due to particulates in the samples, it's one of the reasons why you want to filter your samples. If there is fluorescence or phosphorescence of the sample, if there are changes in the refractive index in terms of the actual concentration of the analyte, if there's a change in the equilibria, or if there's radiation, or if there's stray light. So it's useful to know about Lambert-Beer law, but you also want to make sure that you're taking into account your experimental setups, that you're getting accurate data. One of the reasons why we care about this is if we take a look at the difference between tryptophan and tyrosine. So the absorbance of UV light by tryptophan and tyrosine is one of the ways to tell the difference between amino acids and peptides. If you take a look at our x-axis as opposed to our y-axis, so this is the wavelength of light, absorbance is what we're looking at, and we can actually see a difference then in the behavior of tyrosine versus tryptophan. And if we take a look at the structure then of these molecules, that might give us some clue as to why. So each of these amino acid side chains can absorb UV light. That's because they have this conjugation that exists within the aromatic ring. But they're going to absorb light differently because of the way that light would travel around this ring structure. If we take a look at other types of amino acids, we can actually see here our nonpolar aliphatic R groups. So again, right, we're seeing predominantly carbons and hydrogens. All of these are going to result in nonpolar interactions. Obviously, though, these molecules are going to occupy space differently. And then if we take a look at our polar uncharged R groups, so those are molecules like serine and threonine that have this hydroxyl group, or cysteine, which has the sulfur or the mercapto group, as well as asparagine and glutamine. And these amino acids all have the ability to form hydrogen bonds, and cysteine has the ability to form disulfide bonds. And then if we take a look at our positively charged R groups, we can actually see that lysine, arginine, and histidine, right, all are molecules that have an ability to be deprotonated and lose that charge, depending upon pKa. Then if we take a look at our negatively charged R groups, so aspartate and glutamate, these are carboxylic acids that once deprotonated form this negative anion. So if we think about each one of these molecules, it's reasonable for us then to be able to group into categories, whether those are alkanes or aromatics, carboxylates or amides, positively charged, sulfur-containing, or hydroxyls. And you'll need to have the facility to be able to kind of sort and group these molecules so that you have some sense of how it is they're going to behave or what it is they're going to do. You're going to notice that we always see this Zwitter ion behavior, and it's not always, it's just the most predominant form. So it's possible for this negative charge to be protonated and for this carboxylic acid to be protonated, in which case this molecule would have a positive charge of +1. If you were to deprotonate one of those hydrogens and leave the other one protonated, well, then it would become a charge of 0. If you were to deprotonate both, then it becomes a charge of -1. And if you were to deprotonate the amino group, then it would become a negative charge of 3. So what we're going to be looking at in our next section is how these groups ionize and how they might behave. So we'll come back shortly. Thanks. Have a great one.